Hello everybody, my name is Phil Pinter and today I'm going to talk about phenylketonuria, more commonly known as PKU. So what is PKU? It is a heritable genetic disorder that results in an inborn error of metabolism. Inborn meaning that you are born with it, and error of metabolism is referring to the resulting genetic phenotype that affects the metabolic process. In the case of PKU, individuals cannot digest the amino acid phenylalanine, and the results are toxic. If left untreated, PKU will lead to permanent and severe intellectual disability. It may also result in seizures, growth failure and microcephaly, behavioral or psychiatric problems, hypopigmentation, and most interestingly, a peculiar smell. Body fluids such as urine and sweat, or even hair, have been described to have an odor that is musty or mousy. Perhaps the most interesting thing about PKU is the history of the disease and its discovery, as well as its significance in genetic and medical science. It all started with a Norwegian woman named Borgny Egeland, who had 2 children that suffered from substantial developmental delays. In 1934, the older child was 6 and could only say a few words and barely could walk with spastic movements. The younger child was 4 and could not speak nor walk or even eat or drink on his own. Her otherwise healthy babies had progressively worsening mental disabilities, and no one had an explanation for as to why. In this era, their condition was dismissed as feeble-mindedness. A chance meeting with Asbjørn Folling, a doctor with a background in chemistry and metabolic medicine, led to him agreeing to examine the children. As like others before him, the exams were unremarkable except for the obvious feeble-mindedness, as well as that peculiar-smelling urine. Folling tested the urine samples in his makeshift personal laboratory, and everything came back negative. However, unlike those before him, he added drops of ferric chloride solution to the samples, and something remarkable happened. Normally this test detects acetoacetic acid and turns a purplish burgundy color, but this time it turned a deep green color, a reaction that no one had ever seen before. So 22 liters of urine samples later, Folling had identified the culprit chemical, phenylpyruvic acid. This is how phenylketonuria eventually will get its name. Folling proceeded to test 420 psychiatric patients and found that 8 of them had tested positive and therefore had PKU, which was the cause of their deficit. He published a paper that year that hypothesized an inherited error in metabolism of the amino acid phenylalanine. It wasn't until 1954 that Bickel and Gerhard published the first study that found a dietary treatment for PKU. This led to the first ever medical food, lofenalac, a formula that is free from phenylalanine. Interestingly, phenylalanine is an essential amino acid, meaning that it must be sourced in the diet as the body cannot make it, and even those with PKU who can't tolerate it still require it. So, as the earliest PKU dietary studies showed, if phenylalanine is completely removed from diet, eventually the bodies will break down their own proteins, and phenylalanine will again be present in the urine. Anyway, PKU studies resulted in a lot of scientific firsts. This includes first dietary therapy for metabolic disease, first medical food, and then later, as we'll discuss, the first routine use of newborn screening. So in addition to a lot of these firsts, it had contributed to the understanding of metabolic chemical pathways, biochemical pathways, the relationship between genes and proteins, and it played a substantial role in the birth of genetic medicine. So let's touch briefly on epidemiology. The incidence of PKU is highly variable. The highest incidence is 1 in 2,600 births in Turkey, while the lowest is only 1 in 125,000 births in Japan. These differences are largely attributable to population consanguinity, founder effect, and genetic drift. According to the National PKU Alliance, there are 16,500 people in the U.S. currently living with PKU. Among Caucasians in the U.S., the incidence is somewhere between 1 in 10,000 and 1 in 15,000. There is some definite racial or ethnic disparities as well, as it is more common in Native American or Northern European peoples and less common in African Americans or Japanese. So what's going on behind the scenes here? PKU is caused by a mutation of the PAH gene on chromosome 12. PAH is about 79,000 base pairs, including 13 exons that code for a strand of RNA and a 452 amino acid protein. This protein is the enzyme phenylalanine hydroxylase. As you can see, this enzyme converts phenylalanine into tyrosine, which is another amino acid. It turns out that high levels of phenylalanine has toxic effects on the nervous system, which is why we see intellectual disability as the most significant problem. So studies have revealed that phenylalanine causes hypomyelination and demyelination of nerves. As we know from our friend Dr. Falling, it is also responsible for the characteristic smell. In addition to the direct effects of phenylalanine accumulation, we see a deficiency in tyrosine, which is an important precursor for melanin, the deficiency of which results in the hypopigmentation that we see. Tyrosine is also necessary for the synthesis of thyroid hormones and catecholamine neurotransmitters. Another interesting thing we see with PKU is the phenotypic variability. While it is a biallelic system that follows autosomal recessive inheritance patterns, there is a wide variety of mutations discovered, more than 500, that result in a phenomenon called compound heterozygosity. This results in a range of phenotypes from classic PKU, where there is no PAH activity whatsoever, to what is called variant PKU, where there is some PAH enzyme, and it's present in much smaller levels with much reduced activity. So again, PKU follows an autosomal recessive pattern of inheritance. So if an individual inherits one normal allele from their father and one mutant allele from their mother, they would be a carrier, but would be phenotypically normal and unaffected. However, if an individual inherited a mutant allele from their father and a mutant allele from their mother, they would have the disease. In other examples of autosomal recessive inheritance, we would refer to the offspring as being homozygous recessive because both their alleles have identical mutations. However, in PKU, an individual could inherit a mutant allele from their father and a different mutant allele from the mother, one from over 500 possible mutations. And this is what we would call compound heterozygosity, where two different mutant alleles are present and result in a highly variable phenotype. So how do we diagnose PKU? Well, that's easy. You're born with it and you're tested for it with normal, routine newborn metabolic screening tests. Newborn metabolic screening is done by lancing a newborn baby's heel, placing drops of blood onto paper cards where they are allowed to dry and then sent to state laboratories where they undergo a series of tests for a number of inborn metabolic disorders. Newborn metabolic screening was invented by Robert Guthrie in the 1960s and was implemented in several countries soon thereafter. It is now common practice globally and in all 50 states. Guess what condition was the first to be tested for newborn screening? That's right, PKU. In fact, even to this day, the newborn metabolic screen is commonly referred to as the PKU screen. Beyond newborn screening, blood testing can be done for phenylalanine levels at any point, and abnormal levels can indicate a problem with PAH gene and therefore possibly PKU or some variation of it. Additionally, gene sequence analysis can be performed to determine the exact PAH genotype. The good news is that while it's probably not convenient, definitely not convenient, PKU can be treated and people with PKU can live normal lives without significant symptoms. Simply, those with PKU have to restrict their dietary intake of phenylalanine by avoiding protein-rich foods, taking amino acid supplementations, and consuming medical foods that are 100% phenylalanine free. For those with variant PKU, there is limited but existent activity of the PAH enzyme. There is a medication, tetrahydrobiopterin, that goes by the trade name Kuvan, that is a cofactor for the PAH-mediated reaction that boosts the conversion of phenylalanine to tyrosine. As of so far, there is no true cure for PKU. However, there are some emerging therapies that are really promising. Pegvaliase, or PegPal, is a replacement enzyme that is undergoing phase 3 clinical trials and performs the same function as phenylalanine hydroxylase, or PAH. The next emerging therapy is essentially a cure for the condition and involves gene therapy with a recombinant adeno-associated virus vector that inserts the normal human PAH gene. So far, it has been limited to mouse models, but has had promising results. For those who have PKU or have a family history of PKU and want children, prenatal testing is possible for PKU using chorionic villus sampling or amniocentesis. Parents using in vitro fertilization can also use pre-implantation genetic diagnosis, or PGD, to check for any mutant PAH genes. This is especially relevant for those who have a known history of PKU but don't necessarily have it themselves. It is also important to note that if a mother with PKU is pregnant, it is especially vital to strictly regulate the blood phenylalanine levels, as it is teratogenic. So I'll end this presentation with a familiar image. Anyone who has ever had diet soda has likely noticed this warning on the label. The primary artificial sweetener found in diet sodas is aspartame. Aspartame is a methyl ester of the dipeptide of L-aspartic acid and L-phenylalanine. So consuming aspartame is obviously a big no-no for those with PKU, as it is essentially about 50% phenylalanine. Thank you for your time, and I hope you enjoyed learning about phenylketonuria.